I'm Jayden, a master's student at Washington State University here. My advisor is Dr. Gulhan Unlu. My project here is correlating the microbiome of aged white cheddar cheese to the proteolysis and sensory characteristics during ripening. So, a little about me. I've gotten to come to a couple build events now. Thank you, Kim and Dr. Bastian. But I haven't gotten to present. This is my first time. So, a little bit about me. My parents were in the military, so I grew up all over the United States, but we call Colorado home. Bottom there is a picture of me and my parents in Canada at Yoho National Park. And then above that is my family with my brother and his dog there at the Great Sand Dunes National Park in Colorado. I had horses growing up, and I was on the equestrian team. I was president of the WSU equestrian team for a couple of years, my undergrad. So there's one of my horses there. Most importantly, my cat Sylvie. I will let her know that you all say "Ps, ps, ps." She's the love of my life, and lots of cheese making this year. So lots of fun things. Digging into my project, talking about cheddar cheese here. It plays a major role in the global and U.S. dairy trade. Sorry, I got these little boxes here. The average American consumes 10.9 pounds of cheddar cheese a year. That was from 2024, and that's second in consumption to mozzarella, just because of our love of pizza. Within the United States, revenue for cheddar cheese in 2025 was $13.5 billion, and we are the largest producers of cheddar cheese in the world. So why is that here? Its popularity stems from its flavor, aroma, and texture that's developed through biochemical processes during ripening by lactic acid bacteria, specifically glycolysis, lipolysis, and proteolysis. So bear with me as I explain this little plot here, but I really do like what it's showing. I'm gonna get rid of that. It's a complexity relatedness plot for cheese specifically. This is all of cheese from the Observatory of Economic Complexity. This is for 2024. It's pretty much just comparing a country's potential export opportunity to the strategic value of producing that product. So on our x-axis, that relatedness line moving to the right represents a country's likelihood to develop that new product. And then the upwards on our y-axis is the increase in the country's complexity for productive capabilities. And then the size of the flag there is the amount of production. So I wanted to highlight us here, the United States, and that little green circle for production of cheese. And I recently learned that most of our cheddar that we're importing into the United States is from countries Ireland, New Zealand, and the UK, thinking of the popularity for Irish cheddars, British cheddars, etc. And so we want to be able to compete with those countries and stuff and keep the cheese here in the United States. Similarly, large cheddar and Gouda producers being like the Netherlands and Germany. But I also wanted to highlight countries that are increasing their cheddar cheese or just cheese consumption in general. So looking at like China, India, Indonesia, I think there's a couple others tucked in there, like the Philippines and stuff. So producing a comparable product to some of these other large cheddar producers so that we can stay competitive in the industry here. So looking into some of those biochemical pathways, most importantly being proteolysis for producing flavor and texture compounds. This figure here is specifically for lactic oxide lactase. If you look, notice the PRTP proteinase there. So specific to lactase, but it's generally similar for all lactic acid bacteria. You have primary proteolysis, where those cell envelope proteinases are acting on those casein peptides or proteins, making them into larger oligopeptides, some of those bitter peptides going through into the lactic acid bacteria through the cell membrane, through those peptide transport systems, and going through secondary proteolysis to further degrade those peptides into amino acids and then into amino acid catabolism. So producing what is desirable in cheese without overproducing some of those defectual flavors and textures that can come along with aged cheeses, like bitterness, which can be beneficial, but in excess, it is a defect. So how do we manipulate this? Adjunct cultures. This isn't something that's new. The adjunct cultures have been used as bioprotectives and flavor enhancers within cheese for quite some time. Specifically things like preventing bitterness, debittering, or preventing gas blowing and things like that. Some lactic acid bacteria strains are known for their strong proteolytic activity, looking at Lactobacillus helveticus, which is one of the key players here in this project as well. And these traits vary a lot by strain. So that's generally attributed to minor mutations and sequence variations that are altering the enzyme specificity, gene activity, expression of a lot of those compounds that are being produced during metabolism and by the bacteria. And so understanding what's kind of going on at a molecular level there and how it's affecting affecting the flavor profile of the cheese is really important. So digging into the project itself, first objective, characterizing the microbial communities present in white cheddar cheese with emphasis on that starter, adjunct, and non-starter bacteria. Uh, Second objective, evaluating the sensory properties in white cheddar cheese as a function of microbial and biochemical variation. And then evaluating that peptidomic profile in the cheese, kind of as a validation as to what's happening biochemically, with emphasis on those hydrophobic and known bitter peptides. With this overall goal of understanding microbial diversity in cheddar cheese with relation to the sensory relevant flavor profiles and peptide formation, to kind of get a better idea of what's happening in ripening and how to maximize it. So some materials and methods, I just wanted to include a brief little cheese make diagram here. This presentation is going to be a lot of materials and methods and stuff. I'm kind of hoarding data for the annual meeting, so I'm not talking about the same thing over and over. So we did make three cheeses. I mostly wanted to point out here that we took triplicate samples of our raw milk, pasteurized milk, fresh curd and whey, the cheddar curd, as well as the salted curd right before packaging. And those are being stored at -80 for this metagenomic analysis that will be running through all of our cheeses. So we have three cheeses. They're all commercial products, so we're not listing what like starters and adjuncts they were made with at this time. But cheese A being starter one and adjunct one, cheese B being starter two with no adjunct, and cheese C being starter two with adjunct two. And they'll all be subjected in triplicate each month during ripening to a metagenomic analysis, some descriptive sensory analysis, some analytical analyses, and then that peptidomics work. So digging into that, this is just approximate analysis results from the first month of cheese ripening. We're about 6 months in. We'll actually start 6-month analyses tomorrow. Wish me luck on that. And so, but I thought it'd be more fun to present numbers instead of just what we're looking at. So using a FOSS food scan for dairy solid foods, looking at percent fat, moisture, salt, protein, fat, and dry matter, and then as well as taking pH measurements of our cheeses each month. For that metagenomic analysis, doing some microbiome characterizing with 16S ribosomal RNA sequencing, looking at identification as well as percent abundance of the bacteria within cheese. Like I said, targeting that starter, adjunct, and non-starter lactic acid bacteria, as well as some transcriptomics, kind of map out the transcriptome, look at what genes are being upregulated and downregulated. And this will be in coordination with Oregon State University's Center of Quantitative Life Sciences. And that answers our first objective. Looking at the actual sensory properties within the cheese, we're running a trained sensory panel. This started last September. I trained 10 panelists over a 6-week period, just over 20 hours of training, right before that first time point, which was in October of 2025, which was after 1 month of cheese aging. We made cheese in September of 2025. And then they go through about an hour and a half actually of refresher sessions before each consecutive monthly panel. And these descriptive standards were adapted from Dr. Marianne Drake's Cheddar Cheese Lexicon, actually. It came out in 2001. And panelist feedback was provided with the feedback calibration system and SenPack sensory analysis software. And this is answering our second objective, digging a little bit more into it. These are the 20 attributes that we were evaluating for aroma, texture, taste, and flavor, and they're being evaluated on this 15 centimeter line scale with the reference indicated. So like I said, those reference standards for each of the attributes. Some early results from our sensory analysis. This is just months 1 through 4. Like I said, we're about to enter month 6, looking at this principal component analysis. And I kind of just wanted to highlight the groups of cheeses we have here. We have cluster 1, which is more defined by these early aging attributes. Sorry, excuse me. These early aging attributes like whey, cooked, milk fat, butter, things like that. And then moving on throughout aging, cheeses B and C kind of are in this positive direction here, getting much more harder, a little bit more fracturable, but definitely more bitter and developing more of those aged flavors, free fatty acid and umami. And finally, that third cluster being cheese A as it ages. And those cheeses are labeled as like month times cheese, for anyone wondering. But cheese A being much softer and also aging towards those later aged attributes. For some analytical analysis, we'll do some texture analysis with a texture analyzer. We're running a 2-bite compression test under 20% compression with standardized size and temperature samples. And this can be correlated to those sensory results as well. Similar for the electronic tongue, we're running some electronic tongue analysis. Both of these are in triplicate as well. Everything I should say is in triplicate. There's seven sensors for our samples. They'll go through salty, sweet, sour, bitter, umami, spicy, and metallic. And these results can also be correlated to some of this sensory data from the panel. And then looking at some validation and peptidomic analysis, these peptides are taken from the cheeses in triplicate each month and isolated here at WSU and saved as like an anhydrous peptide. And then they'll be taken to Oregon State along with those metagenomic samples and run in Dr. Dave Dallas's lab using his UPLC equipment and a bitter peptide database in cheddar cheeses, and I believe some other cheeses that was developed by a previous master's student and build student of his, Russell Colefield. So taking advantage of some of that. And that answers that third objective. So current results, lots of cheese. Lots of cheese aging, lots of cheese baking, lots of cheese packaging. Like I said, I'm hoarding data for the annual meeting. Next steps, continue aging our cheese and doing the sensory panels each month, working on sample prep and application of our e-tongue measurements to track that as well. Throughout ripening, perform this metagenomic sequencing to identify key taxa and functional genes, which I'm really excited for. And then analyzing those peptide profiles and stuff and looking at how they relate to the bacteria present and the sensory characteristics in our cheese. So with that, some acknowledgements. My advisor, Dr. Gulhan Unlu, and my lab mates. This wouldn't be possible without them. My committee members, Dr. Carolyn Ross of the WSU Sensory Science Center and her grad students, which have been invaluable, immeasurable help for me, and Dr. Shen of the School of Molecular Biosciences here at WSU. My panelists, for their time and dedication, we're at like 30 some hours of training and analysis now, and we have much to go. I think we get close to 55 hours, so really bless them. And then obviously Bill Dairy and the Western Dairy Center for funding and supporting my research in coordination with our creamery here at WSU, Dairy Gold, and the Oregon State University, CQLS, Dr. Dallas, his lab, and many of the other students and professors there that have helped me and given me advice on things. So with that, more pictures of cheese, and I think I am done. Any questions? Oh, and there's some references.
Okay, I have a question.
Yes,
So
please.
with all this work, what excited you the most and what keeps you motivated when things get tough and don't go the way you want them to?
Well, we've done a lot. So what keeps me excited is one, cheese. Dr. Unlu had pitched this project to me way back when I was originally looking at graduate programs and stuff like that. And I love cheddar cheese. I feel very, very blessed to be here. I knew what I wanted to do. I knew what I wanted to go in, and it just happened that this kind of fell in my lap. So one, I'm blessed that I love what I do. And when times get when things get tough, for your second part of the question there, I just remember that we're kind of all in the same boat. Not to kind of sound like not to draw out this question here. I'm sorry, we're a little bit over time, and not to
You're
sound like I'm
good.
sucking up, but I love that we have this community, this Build Dairy students. We're working with industry, universities, and stuff like that. Because anytime things do get tough, there's tons of people to ask questions to. Like the collaboration's insane. So like I know that there's a bunch of students at Oregon State that have helped me with a lot of things. I've talked to some students at Utah State that have helped me with things. Professor Dr. Adamer has helped me with a ton. Dr. Dallas at Oregon State has helped me with a ton. So it truly just it takes a village.